Welcome to my poster titled Machine Learning Prediction of Molecular Initiating Events Using Chemical Target Annotations and Gene Expression. This work was led by Joseph Bundy and is presented by me, Logan Everett. The bulk of this work was recently published in Biodata Mining and can be accessed using the QR code at the top right of the poster. Please note that the views expressed are those of the authors and do not necessarily represent the views or policies of the US EPA. A molecular initiating event, or MIE, is the initial event triggered by a chemical exposure in a biological system, such as an interaction with a specific protein. MIEs are part of a larger cascade of events called an adverse outcome pathway that can lead to a specific toxicity hazard. The primary goal of this work is to predict MIEs from high-throughput transcriptomics data collected from in vitro chemical screening studies. Here, we combine the Lynx L1000 CMAP gene expression data, specifically for MCF7 cells, with known chemical protein interactions from the RefChemDB resource. We use these to train machine learning models that can predict specific MIEs based on the transcriptional response to a chemical exposure. The first figure on the poster shows our framework for combining these data sources, filtering to appropriate training data, then training and validating the individual machine learning models for MIE prediction. In particular, we collate multiple transcriptional response profiles for multiple chemicals annotated for the same MIE, in this example, estrogen receptor inhibition. We also randomly select response profiles for other chemicals with no known association to the MIE. We trained classifiers using 6 different algorithms: k-nearest neighbors, multi-layer perceptrons, random forest, and 3 different types of support vector machines. All algorithms were applied to 3 different types of input features. All genes refers to the full set of measured and extrapolated genes in the Lynx L1000 data. Landmark genes are only the roughly 1,000 genes directly measured by the L1000 platform. Pathway scores are gene set enrichment scores computed from the all genes set. In all 6 algorithms, the models trained using only the landmark genes outperformed the other input types, demonstrating that for this application, there is no advantage to extrapolating additional genes or transforming to pathway level scores. Because the available training data was limited for many of the MIEs, we performed several analyses to assess overfitting. In addition to comparing internal and holdout accuracy for all models, we also performed empirical significance testing. For each MIE classifier, we created multiple corresponding null models with the same number of chemicals and expression profiles as input data, but with the chemical target annotations permuted from the original RefChemDB data. Thus, each null model is trained using input data with similar structure, including the number of profiles derived from the same chemical, but where there is no meaningful association or shared target for the chemicals in the training set. We repeated training and assessment for 500 null models and used the resulting accuracy scores as a null distribution for the accuracy score achieved by the true MIE model. For each MIE, we removed classifiers that failed this empirical significance test with a p-value greater than 0.05. We further assessed these classifiers on exemplar chemicals that had been completely held out from all training and assessment up to this point. For many of the remaining MIEs, several classification algorithms passed these criteria and were then combined into an ensemble model by averaging the prediction scores from each classifier. This final figure shows the prediction scores for the high-performance classifiers against the corresponding exemplar chemicals. The columns correspond to classifiers, with the MIE labeled at the bottom, and the rows correspond to the exemplar chemicals. Cells marked with an asterisk denote the exemplar chemical for each MIE, and the shading indicates the percentile rank of the prediction for that classifier against that chemical. All exemplar chemicals shown here were among the highest ranked predictions for each MIE classifier. However, there are also cases where MIE classifiers return a strong prediction on other exemplar chemicals. These may represent potential crosstalk between the molecular targets. In conclusion, this work represents a proof of principle application of machine learning to the task of predicting MIEs, such as chemical protein interactions, based on transcriptomic response profiles. In particular, we found that we could predict 11 MIEs with high confidence using the available data from a single cell type, MCF7, and the existing chemical target interaction data for training. Furthermore, we found that the best performance was achieved by limiting the input features to only the measured genes on the L1000 platform. Thank you for visiting my poster, and please let me know if you have any questions.